Section 2. Secondary messaging, crosstalks, downstream. Please don't panic at my first overview slide, which I'm about to take you through a bit later. I want to focus your attention on one thing first. It's an incredibly complex network of molecular interactions. The discussion about the PIPs and the PTDN cycle from section 1 also leads me now seamlessly into this section problem, secondary messaging. So we have just learned that PIPs and PTDNs play one of the most important regulatory roles in intracellular processes and, in addition to regulate membrane structure and the uptake of substances via endocytosis, send something downstream, signaling that further processes are needed. I would like to divide this complex into three sub-aspects. What do we mean by secondary messaging and signal transduction cascades? What do these cascades do? And why do we have a serious problem if we change the activation patterns and expression profiles of these cascades out of context? So, what does secondary messaging have to do with signal transduction cascades? We define cascades as downstream interactions between individual signaling molecules which are understood as a canonical sequence. Why do I say understood? Because there will, in a typical interaction pathway, no canonical signaling pathways in reality. Imagine a cell as a highly organized, smallest living unit that communicates through specific signaling molecules. These molecules activate signaling pathways called cascades because they trigger a series of reactions that go further and further downstream. Post-translational modifications, such as phosphorylation or ubiquitination, which alter proteins and thus regulate their function, are a central mechanism in these processes. This enables precise control of the cell response. Signaling cascades are not only linear, but often run in parallel with the pathways influencing each other and thus enabling a finely tuned and very tightly regulated cell response for each aspect you can imagine. These cascades usually end in the cell nucleus, where they regulate the activity of genes. In other words, transcription and translation. What still sounds very theoretical here, I hope will make much more sense with my slide. We will go through this slide step by step. We have signal cascade X and signal cascade Y. We first follow the black arrows of cascade X. This would be the canonical linear pathway. We assume that the first molecule is activated by a typical receptor interaction. The first molecule releases one phosphate to the signaling molecule connected after it, thereby activating it, which leads to the next molecule. This in turn releases 3-phosphate to the next molecule, which can then enter the nucleus and gives 2 phosphates in an activated state to the target gene, A. A similar process takes place with the canonical form of cascade Y, but cascade Y is working with ubiquitination. Now we follow cascade X again, but first we look into the starting molecule of cascade Y. Cascade Y shares one ubiquitin with the downstream molecule from cascade X. The rest of the cascade X continues as normal, or maybe another effect maybe can observe would be that it is suppressed, so inhibited, amplified, or attenuated. I think that if this principle is understandable up to this point, the rest of possible crosstalks, which should only be a schematic example in the slide, can be logically understood. I would have liked to give more examples of the theory behind the crosstalks, but I want to keep this short and as clear as possible. Before we move on and get back to my first slide, which probably hit like a bomb and caused some confusion for some people, I have to mention that my perspective is also limited to only 2 of the 5 forms of signal transduction, as even my brain is starting to boil in this perspective if I would also follow the justacrine, paracrine and endocrine pathways. And it's difficult to separate them cleanly anyway, because every intracellular reaction necessarily has systemic effects on these pathways as well. But now to my slide, which I'm frankly a little proud of, because it's a systemic representation of what all happened in these mod RNA shots even before the spikes come into play, as an additional driver of this broth. We go through from the bottom. Firstly, the LMPs tile onto the membrane and inevitably activate the petidin cycle in a devious and unnatural way, which we looked at in detail in the first section. This inevitably also activates mTORC1-2, the mechanistical target of rapamycin complex 1-2, one of the signal transduction cascades we just discussed in theory, as its PI3K phosphatidylinositol 3 kinase is involved in the regulation of petidins and is activated by RAS, a signaling molecule of MAPK, the mitogen activated protein kinase. PI3K is the effector protein, RAS the activator. And since cascades always activate the next downstream protein, we have already unbalanced two huge and almost impossible to manage signaling cascades. This will end in the activation of ERK and ACT signaling molecules, ERK for the MAPK, ACT for the mTORC, which then enter the cell nucleus. This already takes us to step 4, while step 2 is taking place at the same time. And this, you have to understand, this runs parallel and at the same time. Don't get too confused about all the green cyclic arrows in the slide, cause they are just showing, which I discussed also in my substack Downstream Teil 1 to 7, in much more detail, the crosstalks between 4 huge signaling cascades, mTORC, CAMP, JAK-STAT, and MAPK, which are directly involved in this issue. In other words, while the endosomal escape is being regulated, step 3, transcription factors are already being activated, as this is a highly inflammatory process and stress factor for the cell, and it has to react to it. So the whole transfection is already highly inflammatory. ERK and ACT will activate transcription factors and completely shake up gene expression as a highly inflammatory and stress-related process is taking place. Transcription factors are special proteins in the cell nucleus that are the target of signals from the cell, which are transmitted through various cascades. So imagine them as the molecules inside your nucleus that switch on or start transcribing genes. Transcription factors can be taught as switches that are flipped precisely when the cell needs a particular gene. Several transcription factors often work together to precisely regulate the speed, duration, and amount of gene activity. This ensures that the right genes are active at the right time and in the right place. In other words, while the endosomal escape is still being regulated simultaneously by these kinases, a special form of our cascades, and other signal transduction pathways, step 3, transcription factors are also already being activated, as this is a highly inflammatory process and stress factor for the cell, and it has to react to it. And then comes step 5, translation after the endosomal escape and the regulation of the mod RNA. Here too our cascades are involved to activate and regulate microRNAs and ribosomal regulatory proteins. Ribosomes are the power machines required for translation, in other words, for translating the mod RNA and folding it into a 3-dimensional protein structure. MicroRNAs are therefore to regulate the translation when it's necessary. And a little joke to boot. We have just skipped the step that is normally also regulated between the endosomal and lysosomal phases, which is already leading to acute malfunctions in the tightly meshed regulation by our cascades. Because this crappy lipid nanoparticles will therefore rupture the endosome. So to summarize the damage potential of LMPs, which therefore I'll discuss in detail in section 3 and answer my third question. And why do we have a serious problem if we change the activity patterns and expression profiles of these cascades out of context? Let me use an analogy. Imagine a three-dimensional gearbox consisting of countless gears. Each individual tooth must mesh with absolute precision. And now imagine that you have worn down a few teeth to such an extent that the gearbox will continue to run, but the damage can no longer be compensated for. Can you hear it already? Click, click, grind, click. And now imagine this three-dimensional gearbox as a complex network of signaling transduction in our cell. Every reaction and every molecule has its specific place and time at which it takes action to initiate or regulate processes. This is how the cell works. This is how your body functions precisely like a well-coordinated clockwork. Each cell has its own program, and the real wonder is how they work perfectly timed together, forming all higher processes including your thoughts and feelings. Now, if some gears, let's say proteins or signaling components, are worn or damaged, they no longer work precisely. There is a small offset in this process, a click here, a grind there. In the beginning, the system may still work, even if the processes are not completely smooth. But deviations add up. Over time, the system wears itself out, the damage increases, and the small faults lead to larger ones. In the long term, this can throw the entire system out of balance. In signal transduction, this would mean that misregulation or incorrect activation occurs in important cell processes such as cell division, cell survival, and programmed cell death. Over time, cells could begin to divide uncontrollably, a potential pathway towards tumor formation, react incorrectly, which could lead to autoimmune diseases, neuronal degeneration, chronic inflammation, etc., etc., or fail to regenerate properly, which would accelerate wear and tear and promote signs of aging. This click, grind, click is therefore the early warning signal. If these small defects occur too often or in too many cells, the body can no longer compensate for the damage. The result is progressive degeneration, which can be accompanied by chronic diseases and increased risk of infections and ultimately a shortened lifespan. And the most bizarre thing you might not want to think about, it will have systemic consequences that can affect you in such a way that it will also affect your children. This is the epigenetic component. The small, insidious defects can leave profound and long-term changes in gene regulation through epigenetic modifications. This means that the damage not only remains in the affected person, but can potentially be passed on to the next generation. Epigenetic markers are like nodes that the cell leaves on the genetic wiring diagram, I would call it so, and they can include altered instructions for basic functions, cellular stress responses, immune responses, or even susceptibility to certain diseases. Even if the children do not directly suffer the same cell damage, they may show increased susceptibility or altered responses to stress factors. It almost is as if the click, grind, click echoes not only in your body but in your family tree, a kind of epigenetic inheritance that is passed on from generation to generation. And the absolutely most disgusting part, you might not realize it for your own, but it could become even more potent to the next generation. And a brief closing remark for this section. The literature behind my slide and my explanations is so long that it is impossible to list everything. The links to the key studies I based this on can all be found in my downstream parts 1 to 7, so in my substacks.